Welcome to our seminar. My name is Erika Leonard and I'm the Director of Quality Control and Senior Research Histologist at Vector Laboratories, and this seminar will be considerations when designing your immunohistochemistry procedure. In the last section, we discussed optimizing your protocol to ensure optimum performance of your detection system, your primary antibody, and your detection system, as well as performing deletion controls to determine sources of extraneous backgrounds. In this section, we'll discuss advanced detection protocols involving multiplexing and species-on-species detection. Multiplexing is detecting multiple antigens simultaneously, and species-on-species detection involves using this primary antibody of the same species as your specimen. Our first topic is multiplexing. What is multiplex and why would you want to do it? Multiplexing allows you to maximize the information you can obtain from a single specimen. It can demonstrate spatial relationships of specific target antigens, and it can help you to identify co-expression of multiple antigens. You'll need to decide whether you want to do your multiplexing immunohistochemical procedure using chromogenic or fluorescent reagents. Both staining methods can be used quite successfully, and there's many color choices available for each of these. However, if the two target antigens are co-localized in the same compartment of the same cell, you're probably going to want to choose a fluorescent detection system over the chromogenic detection. In this schematic, you can see a chromogenic multiplexing using our Impress reagents. Impress reagents are ideal for multiplexing because they're a one-step, ready-to-use solution which provides high sensitivity and low background. Following your first primary antibody incubation, you'll incubate with your Impress reagents for 30 minutes, followed by your enzyme substrate. Following the enzyme substrate step, you'll apply your second primary antibody, followed by your second Impress reagent and your second enzyme substrate. There are some important considerations when undertaking a multiplexing procedure. The first thing is to make sure that you optimize your single label protocols first. You'll want to ensure that your signal-to-noise ratios are optimal for each of these single label protocols before combining them into a multiplexing procedure. Second of all, before you undertake your multiplex procedure, you're going to want to design and diagram the entire protocol to ensure that there's no cross-reactivities or incompatibilities with any of the reagents that you have chosen. Your primary antibodies, substrates, fluorophores, counterstains, and mounting media must all be compatible for you to have a successful multiplexing experiment. The third thing you need to do is you'll want to choose your reagents. Impress polymer reagents are ideal. There's minimal steric hindrance because there aren't the multiple components required in a two-step procedure. You'll want to determine the optimal sequence to apply your primary antibodies, and you'll want to run a series of controls to compare your single label results to your multiplexing, and also deletion controls to make sure you don't have any background or crosstalk between your detection reagents. If you're doing a chromogenic co-localization, most combinations of substrates, when they're deposited at the same site, do not produce a third color. There are some exceptions to this rule. Vector red AP substrate and vector blue AP substrate produce a third color if deposited at the same location. This is also true of vector nova red and vector blue. In this schematic, we're showing a fluorescence multiplexing using primary antibodies from different species. After we incubate with our first primary antibody, a biotinylated secondary antibody is added, which detects the species of the first primary. We add a fluorescein avidin DCS to detect that secondary antibody, followed by an avidin biotin block to block any excess avidin or binding sites available. We then add our second primary antibody and the second biotinylated secondary antibody. This is then detected by a Texas red avidin DCS, and then we can mount and observe the two fluorophore colors. If you're doing chromogenic multiplexing, a chromogen can develop and precipitate over the detection system, offering a shelter of any exposed antibody epitopes underneath that enzyme system. However, fluorophores are discrete molecules that are conjugated to your detection reagents. Therefore, fluorescent secondary antibodies don't shelter that bound primary antibody. This offers some challenges if doing same species antibodies, because the secondary antibodies can't distinguish between the two primary antibodies made in the same species. So you have several solutions to this problem. One is to change one of the primary antibodies to another species. If the two primary antibodies are both prepared in mouse, you can use our vector M.O.M kit or mouse-on-mouse detection system to detect the second mouse primary antibody. We're now going to move on to the second topic in this section, which is species-on-species detection. There is a challenge when detecting species-on-species. What we mean by this is that the primary antibody is the same species as the specimen, for example, mouse-on-mouse or rabbit-on-rabbit. And your detection reagent will bind not only the primary antibody, but any endogenous IG that's present in your specimen, resulting in a high background signal. There is a species-on-species solution, which is a flexible, adaptable technique which was recently published, which pre-incubates Fab fragments of secondary antibodies with your primary antibody in solution to preform a complex. We then apply that complex onto the tissue section. Vector offers a special solution for mouse-on-mouse situations, which are called the Vector M.O.M Immunodetection kits. They're able to localize mouse primary antibodies on mouse tissue, avoiding the extra background due to endogenous mouse IgG. We offer a unique mouse IG blocking reagent which minimizes the undesired binding of the secondary antibody in your detection system to the endogenous mouse IgG. Mouse-on-mouse detection is extremely important because mice are so widely used in model systems, xenografts, and other applications, and we've developed this unique solution specifically for this challenge. As you can see in this example, without using the Vector M.O.M mouse IG block on the left, a significant amount of endogenous mouse IgG is being detected by our secondary antibody. After using the Vector M.O.M mouse IG block, we're able to see the specific staining of the actin in this mouse kidney. In this section, we discuss two advanced protocols. With careful selection of detection strategies, dyes, and protocols, chromogenic and fluorescent multiplexing can deliver valuable information. And when using a primary antibody of the same species as the tissue specimen, the detection secondary antibody will bind to both the primary antibody and endogenous IG, causing high background. The Vector M.O.M immunodetection kits solve this challenge for mouse-on-mouse situations. Thank you for joining us for our seminar, and I invite you to listen to the additional sessions which are available on our website.